The fluid mosaic model was proposed by S.J. Singer and Garth L. Nicholson. This model explains the structure of the plasma membrane of animal cells as a mosaic of components such as phospholipids, proteins, cholesterol, and carbohydrates. These components give a fluid character to the membranes. What is it made up of? The cell membrane is primarily made up of three things. They are namely phospholipids, cholesterol, and proteins. Now let's see about phospholipids. A phospholipid has two main parts, the head and two tails. The head is a hydrophilic phosphate molecule attracted to water. The two tails are composed of fatty acid repel water ropae. Because of their hydrophilic and hydrophobic characteristics, phospholipids are organized into two layers as cellular membranes develop. The phosphate heads in each layer face the aqueous or watery environment on either side, and the hydrophobic tails hide away from the water between the layers of heads. Moving on to cholesterol. As it is present between phospholipids, it prevents the compaction of hydrophilic tails at low temperatures and their expansion at high temperatures, thus maintaining the fluidity of the plasma membrane. Proteins. The plasma membrane has three types of proteins: integral proteins. These proteins form channels to allow the movement of large molecules and ions across the hydrophobic layer of the membrane. Peripheral proteins. These are found embedded in a single leaflet of the membrane. They carry signals from one segment of the membrane and relay it to another. Glycoproteins. They stabilize the membrane and are responsible for intercellular communication. What makes up the cell membrane fluid? Lipid rafts. These are the lipid domains found on the external leaflet of the plasma membrane. Cholesterol, glycosphingolipids, and glycosylphosphatidylinositol are the building blocks of lipid rafts. Protein complexes. Proteins and glycoproteins are diffused within the plasma membrane. These help in the transport of ions and metabolites, cell signaling, adhesion, and migration. Restriction to fluidity of plasma membrane. Temperature affects how the phospholipids move and how close together they are. When it's cold, they are found closer together, and when it's hot, they move farther apart. Cholesterol molecules are distributed randomly along the phospholipid bilayer and prevent it from separating too much or compacting too much. Saturated and unsaturated fatty acids. Phospholipid tails are made up of fatty acids. Saturated fatty acids contain only single bonds between carbon atoms. Thus, the chains are straight and easy to pack tightly. Unsaturated fats are chains of carbon atoms that have double bonds between some of the carbons. The double bonds create kinks in the chains, making it harder for the chains to pack tightly. The plasma membrane comprises amphiphilic phospholipid molecules. The second important component of the plasma membrane is integral proteins that are integrated completely into the membrane. Carbohydrates are found on the external surface of the membrane, where they are bound to proteins or lipids. Thank you all. Don't forget to like, share, and subscribe